Hello everyone. Today I will be speaking on various disinfection methods. Disinfection methods can include sterilization and the action of disinfectants. Sterilization kills all measurable agents. A disinfectant is a physical or chemical agent that destroys pathogens but does not necessarily kill them all. They're expected to kill more than 99.999% of the test organisms, usually applied to water and inanimate objects or fomites, foods, and aerosols. A bacteriostat is usually a chemical agent that prevents the growth of bacteria, but does not necessarily kill them. I will be discussing thermal disinfection, some of the factors affecting the use of disinfectant, and commonly used physical and chemical disinfection types. Thermal disinfection employs elevated temperatures to inactivate or kill pathogens, a fundamental approach in both historical and modern disinfection practices. The underlying mechanism involves the denaturation of microbial proteins and nucleic acids, disrupting cellular integrity and function. Techniques range from pasteurization, which operates at temperatures between 60 Celsius and 85 Celsius, and used to reduce pathogens in food and beverages, to sterilization processes in medical settings, where temperatures often exceed 121 degrees Celsius under pressurized conditions. The efficacy of thermal disinfection is a function of both temperature and exposure time, evaluated by measures such as the D value, which is the time required at a specific temperature to reduce the microbial population by 90%, and the Z value, which is the temperature that achieves a 10-fold reduction of the D value. The kinetics of disinfection provides a quantitative framework for understanding how rapidly pathogens are inactivated by disinfectants. It is usually modeled by first-order kinetics, where the pathogen death rate is proportional to the number of pathogens. Key parameters include the concentration of the disinfectant, the contact time, pathogen characteristics, pH, and temperature. Advanced kinetic models may incorporate factors such as pathogen resistance and the presence of organic matter in the environment, providing a more comprehensive understanding essential for optimizing disinfection protocols in clinical, industrial, and environmental applications. The figure here is a general model for infection dynamics. For example, while the expected death rate from the model is linear, the blue line, one can see a shoulder curve, the red line, which may indicate the presence of clumps of organisms or multiple hits of critical sites before inactivation. Halogens, including chlorine, chloramines, chlorine dioxide, bromine, and iodine, are potent chemical disinfectants used in water treatment, healthcare, and sanitation. They inactivate microbes through oxidation and disruption of cellular processes. Their effectiveness varies with pH, temperature, and organic matter presence. Chlorine is widely used in water treatment, forming hypochlorous acid, HOCl, in water to disrupt cellular structures of microorganisms. It is effective against a broad range of pathogens, but can form harmful byproducts like trihalomethanes and is less effective in the presence of organic and inorganic materials. Chloramines, formed by chlorine and ammonia, provide a longer-lasting residual disinfectant effect, especially in distribution systems. They form fewer harmful byproducts but are less potent than chlorine, requiring longer contact times and careful balance of chlorine and ammonia concentrations. Chlorine dioxide disrupts protein synthesis and cell permeability, effective against a wide range of pathogens, including chlorine-resistant ones. It produces fewer byproducts and is used in drinking water, food processing, and healthcare. However, it requires careful on-site generation due to its instability and safety concerns. Bromine and iodine are less commonly used halogens but offer specific advantages. Bromine, often combined with chlorine, is effective in high-temperature environments like spas and pools, with efficacy less affected by pH fluctuations. Iodine, used in medical antiseptics and emergency water disinfection, is effective against a wide range of pathogens. Both can form bioactive compounds, necessitating management to mitigate health risks from long-term exposure. Ozone is a powerful oxidizing agent used in water and air disinfection. It generates reactive oxygen species that cause irreversible damage to microbial cell walls, membranes, and genetic material. Ozone is effective against bacteria, viruses, and protozoa, including chlorine-resistant species. Its application in water treatment results in minimal formation of harmful byproducts. However, ozone is highly reactive and unstable, necessitating on-site generation and careful control to prevent exposure to humans and animals. Its short half-life also limits residual disinfection, requiring integration with other methods for comprehensive microbial control. Many metal ions have antimicrobial properties, but because of toxicity, only silver, copper, and zinc have found much use for disinfection. These ions interact with microbial cell components, causing structural damage and interfering with essential enzymatic processes. Silver ions are particularly effective in medical devices and wound dressings due to their broad-spectrum activity and low toxicity. Copper and zinc are used in water treatment and agricultural applications. The efficacy of metal ion disinfection is influenced by factors such as concentration, exposure time, and the presence of materials that may interact with the ions and reduce their availability. Ultraviolet, or UV, disinfection employs UVC light, a higher frequency UV light, to inactivate pathogens by causing damage to their DNA and RNA, preventing replication and leading to cell death. UV disinfection is widely used in water and air treatment, providing rapid and effective microbial control without the formation of harmful byproducts. The efficacy of UV disinfection is dependent on factors such as wavelength, intensity, exposure time, and water clarity. UV systems must be carefully designed and maintained to ensure consistent performance, and are often used in conjunction with other disinfection methods to achieve comprehensive microbial reduction. Photodynamic inactivation, or PDI, involves the use of photosensitizing agents that, upon exposure to light, produce reactive oxygen species capable of destroying microbial cells and viruses. This method has shown promise in treating infections and disinfecting surfaces. Photocatalysts, such as titanium dioxide, generate reactive species under UVA light, a longer wavelength UV light with less toxicity, offering another avenue for effective pathogen microbial inactivation. These advanced techniques hold potential for applications in healthcare, water treatment, and environmental remediation. The effectiveness of PDI and photocatalysts is influenced by factors such as light wavelength, intensity, and the nature of the photosensitizing agent or catalyst. Gamma and high-energy electron irradiation utilize ionizing radiation to disrupt microbial DNA and cellular structures, rendering them non-viable. Gamma rays, produced by isotopes such as cobalt-60, penetrate deeply into materials, making them suitable for sterilizing medical devices, pharmaceuticals, and food products. High-energy electron beams offer similar disinfection capabilities with shorter processing times. These methods are highly effective against a wide range of microorganisms, including spores and viruses. However, their use requires stringent safety measures to protect operators and the environment from radiation exposure. Other chemical disinfectants in common use include quaternary ammonium compounds, or quats, like benzalkonium chloride, which are often used in antiseptic products, triclosan, a mild-to-the-skin bisphenol that is often used in hand sanitizers, and ethylene oxide gas to sterilize heat-sensitive equipment. In conclusion, the array of disinfection methods available today provide versatile tools for controlling microbial contamination across various settings. From thermal and chemical approaches to advanced physical techniques, each method offers unique advantages and challenges. Understanding the mechanisms, kinetics, and factors affecting each disinfection strategy is crucial for optimizing their application and ensuring effective microbial control. Thank you for your attention, and I look forward to any questions or discussions.